The standard advice for sleep hygiene is highly consistent. Turn down the thermostat, set the room to a cool 68 degrees, and leave it there for the night. We treat a static, cool room as the absolute biological ideal for rest. But mammalian thermoregulation does not stay static across the night. Instead, our body's ability to manage its own temperature fluctuates dramatically depending on the specific stage of sleep architecture we occupy at any given moment. A 2025 clinical study by Kim and colleagues hypothesized that sleeping on a surface with a constant temperature actually causes sleep fragmentation. By holding the environment static, we fail to meet the shifting biological needs of the brain. This flowchart illustrates the study's crossover design, showing the path from natural sleep to constant temperature and real-time adjusted sessions. Researchers monitored 25 participants using polysomnography. This medical test records brain waves, oxygen levels, heart rate, and breathing to map exact sleep stages. This graph shows mattress temperature fluctuating with sleep stages. An AI algorithm monitored breathing sounds to detect sleep transitions, triggering real-time adjustments. During non-REM sleep, the mattress maintained a warm 33 degrees Celsius. During REM sleep, it actively dropped to 30 degrees. The researchers posited that optimizing restorative sleep requires matching the external microclimate directly to the internal state of the hypothalamus. Doing so minimizes wake-after-sleep onset, or WASO. This clinical metric tracks the total minutes a person spends awake after initially falling asleep. The long-held advice of maintaining a cold, static room is physiologically incomplete. Deep, restorative sleep requires an environment that shifts directly in tandem with the brain's thermal vulnerability. To understand why the mattress targets 33 degrees during non-REM sleep, we have to look at the distal to proximal thermal gradient. Peripheral skin warming, specifically at the hands and feet, is the biological trigger that causes the core body temperature to cool down. This is commonly observed as the warm bath effect. Passive skin warming dilates blood vessels, facilitating a redistribution of heat away from the core. Dropping that core temperature is a strict physiological prerequisite for falling into non-REM sleep. The sensory mechanism begins with TRP ion channels in the skin. TRP channels are specialized proteins that detect changes in ambient temperature. When they detect warmth, they send a signal up to the lateral parabrachial nucleus. This is a specific cluster of neurons located in the brainstem that relays sensory information to higher brain regions. From the brainstem, the signal travels to glutamate and NOS1 neurons located in the median and medial preoptic nuclei. These nuclei, abbreviated as MNPO and MPO, are precise regions within the hypothalamus responsible for integrating temperature signals and regulating sleep. Activating this exact cluster of hypothalamic neurons accomplishes two tasks simultaneously. It commands the body to undergo systemic cooling, and it heavily induces the slow-wave EEG patterns characteristic of non-REM sleep. This neurobiology explains the 33-degree mattress setting. It mimics the specific warm microclimate required to keep the skin's TRP channels firing, ensuring the sleep-promoting circuitry in the hypothalamus remains continuously active. We see this across species. Animals utilize nesting and huddling behaviors to build these precise microclimates. It allows them to reach a state of thermoneutrality, where core temperature is maintained with zero extra energy expenditure. Skin warmth operates as a direct neurological command. Providing a 33-degree microclimate satisfies the body's thermal defense requirements, preventing arousal mechanisms from waking you, and allowing the brain to sink deeply into non-REM sleep. But the rules change completely the moment the brain enters REM sleep. During REM, mammalian thermoregulatory defense mechanisms are effectively suspended. The brain enters a state of high vulnerability. It becomes poikilothermic, meaning it temporarily loses the ability to regulate its own internal temperature against external heat or cold. If the sleeping surface remains locked at the warm 33 degrees optimized for non-REM, the poikilothermic brain registers immediate thermal stress. The result is rapid sleep fragmentation and heavily truncated REM episodes. This chart of the algorithm's adjustments shows the solution. During REM sleep blocks, the mattress dynamically drops the microclimate to 30 degrees Celsius to accommodate this complete loss of thermoregulatory capacity. Because the body's internal thermostat is offline, the mattress acts as an external surrogate. This dynamic cooling protects the REM cycle from heat-induced arousal. External cooling is strictly necessary here because REM sleep inherently generates internal heat. During this stage, blood flow shifts, with warmer vertebral blood increasing relative to cooler carotid artery blood, physically raising the temperature of the brain. We lose the ability to sweat or pant effectively during REM. A static, warm environment becomes an immediate biological threat. Dynamic cooling is required to safeguard this fragile, highly active cognitive state. The 2025 clinical trial tested these exact mechanisms, and the resulting polysomnographic data shows measurable, objective improvements across the entire architecture of the night. This bar chart displays the trial's core metrics. The green bars representing the real-time adjusted condition show significant increases in both total sleep time and overall sleep efficiency compared to natural sleep and constant temperature controls. Looking at the third cluster of data, the real-time adjustments generated a distinct reduction in wake after sleep onset, proving the protocol successfully reduced nightly sleep fragmentation. This chart isolates specific sleep stages and latency. Note the massive reduction in the REM latency metric. Dynamic cooling physically accelerates the brain's entry into restorative REM stages. The study also included a subgroup analysis, revealing that male and female physiology responded differently to the adaptive thermal regulation. This chart breaks down the male subgroup. Males experience the most statistically significant gains in their overall REM sleep percentage and sleep efficiency under the active cooling protocol. Conversely, this chart shows the female subgroup data. Females saw their most significant enhancement in deep sleep consolidation, with their deep sleep percentage climbing from 7.1% in the control group to 12.3% under dynamic regulation. Whether driving deep sleep consolidation in women or REM duration in men, matching the external temperature to the internal sleep stage physically restructures the night for optimized recovery. Why does the brain prioritize cooling so aggressively during these specific sleep stages? Cooling down during sleep is not a passive byproduct of lying still. The physical temperature drop is the biological trigger initiating critical neuroprotective housekeeping functions. The lower temperatures encountered during sleep induce the expression of CIRBP and RBM3. CIRBP, which stands for cold inducible RNA binding protein, and RBM3 are specific genes that activate at lower body temperatures to alter clock gene expression, protect neurons, and initiate cellular repair. Static environments fight this natural circadian rhythm. When the room temperature doesn't match the required sleep stage, the brain wastes energy defending its core temperature rather than expressing the genes needed to repair itself. The next frontier of sleep hygiene relies on a responsive thermal microclimate. By mapping temperature to the exact neurological state of the user, the bed effectively operates as a second hypothalamus. Adaptive thermal regulation proves that for the brain to truly rest, the environment must stop being static and start being as dynamic as our biology.